and co-hosted by the Undergraduate Research Society of Purdue and the Office of Undergraduate Research. My name is Preetham. I am the current president of UGRSP, and I am so excited to welcome you all here today. Just friendly reminder, no food or drink are allowed in the auditorium. If you do have to exit or enter at any time, please do so in between the presentations to avoid any disturbances. If you haven't done so already, please fill in the attendance sheet if you are a Purdue student. If you are interested in our club and or joining, please join our mailing list, as that is the best way you will get information from us, and also follow our social medias. Without further ado, let's get started. Tonight, 5 amazing, talented presenters from all disciplines across Purdue's campus are going to be presenting their 3-minute research pitches to an amazing panel of judges and our audience. Afterwards, each judge will be given the opportunity to ask one question to every presenter, in which that they will have to answer in 1 minute. Between each presentations, judges will take time to score and deliberate, and after all presentations are concluded, the judges will determine our winners. But you, the audience, also have a chance to make your mark tonight. You guys have all received a audience vote card as you came in, and you'll be able to vote for your audience favorite presentation. Our first place winners will win $300 and a $50 gift card to the Purdue University bookstore. Will the slider work? There we go. Our first place winner will win $300 and a $50 gift card to the Purdue University bookstore. Our second place winner will win $200 and a $50 gift card to Who Hot Mongolian Grill. Our third place winner will win $100 and a $25 gift card to Bruno's Pizzeria. And our audience favorite will win $100 cash prize. Every presenter tonight will also be going home with tickets and family packs for Monster Mini Golf. Huge thank you to all of our sponsors, and a special thank you to the Office of Undergraduate Research for providing the award money. All right, tonight for you, we have 3 amazing judges with us. First up, we have Dr. Shalini Lonam, who is an assistant professor in chemistry and physics at the Purdue College of Science. Her research is in cellular biophysics with an emphasis on quantitative fluorescence imaging. Her group is focused on measuring spatial and temporal mechanisms of cellular decision-making with single molecule and single cell resolution. They address questions related to the physical mechanisms that drive tumor evolution and the corresponding immune responses. And they strive to generate actionable insights to promote targeted, cell-based cancer therapeutics that take advantage of the potent activities of small numbers of activating events. Providing an interdisciplinary skill set to her trainees and students motivates her. She enjoys the academic environment for its diversity, innovation, and discovery, and especially the curiosity and collegiality. Her husband is an architect, and he constantly inspires her to find both creative and clever solutions to problems. She is also an origami enthusiast and enjoys cooking, painting, and disc golf. Please give her a warm round of applause and welcome. Next up, we have Dr. Tara Saunders. She's a production editor with Purdue University Press. She has particular responsibility for their collection of subscription and open access journals, including JPER, the Journal of Purdue Undergraduate Research. She holds a PhD in U.S. history from Indiana University, and her research interests include the social and cultural history of 20th century America, as well as the history of business and work, the history of gender and sexuality, and the history of material culture and everyday life. Please give her a round of applause as well. And last but not least, unfortunately, our last judge, Wasif, was not able to make it here due to a personal issue. So instead, we have our mentor of our Undergraduate Research Society, J.J. Sadler, and he's also the Associate Director for the OUR. So please give him a welcome as well. All right, without further ado, let's get started. Upcoming is Catherine, who will be your MC for the night, and she will announce our first presenter. Enjoy, everyone.
Hi everyone, welcome to the Research Pitch Competition. Thank you so much for coming tonight. My name is Catherine and I am the College of Science Liaison for UGRSP. And if you'll please join me in welcoming our first presenter of the night, Anaya Edwards, a senior in civil engineering, who will be presenting humidity, a key factor in the properties of recycled textile reinforced mortars. Please welcome them to the stage.
The world is growing quickly, with the global population figure reaching 8 billion toward the end of 2022. The fashion industry and the construction industry, although seemingly two opposite ends of the industrial spectrum, are both trying to accommodate the growing demand of the growing populace. You're probably familiar with the term fast fashion, coined in the early 1990s toward the textile production boom, which today contributes to over 100 million tons of textile waste to our landfills annually. Meanwhile, the construction industry consumes billions of tons of natural resources every year to build our workplaces, our homes, and our schools. So then the question becomes, how do we mitigate the threat of excessive waste from mass production while improving upon our infrastructures with a lack of natural resources? Well, one of my solutions is rather straightforward: building with our trash. The use of recycled textile fibers, or RTF, in the production of cement-based materials enhances the material's properties while reducing the amount of natural resources used. However, little research evaluates the welfare of RTF under varying real-world environmental conditions. My research aims to evaluate the effect of humidity on the thermal and mechanical properties of RTF as a reinforcing material in mortars. This project began rather humbly by hand shredding 2 pairs of used commercial denim pants with a pair of scissors and a blender. The fibers, which we cut to be approximately 0.5 to an inch long, were hybrid fibers made of a combination of cotton, viscose, and polyester. We mixed sand, water, cement, and fibers at volumetric concentrations of 0, 0.5, 1, and 2% to make our mortar samples. For each mixture type, 9 rectangular samples were cast, set in a humid curing chamber, and separated into groups of 3 to be made and tested at 0%, 50%, and 100% humidity levels. All of these were then tested for their thermal properties, bending strength, and compressive strength. The results of our research so far revealed that an increased level of humidity resulted in an overall increased level of thermal conductivity, whereas an increased fiber presence resulted in a decreased level of thermal conductivity, creating a more heat-retaining and energy-efficient material. Furthermore, the addition of fibers in our mortars does decrease the overall strength. However, this is accommodated by an apparent increase in ductility or bendableness, which could be the lifeline between a sudden disastrous failure or a gradual warning crack. These results reflect the complexities of the textile itself. It may seem like we're regressing to a more simplistic means of innovation by using waste to build our infrastructures, but RTF as a reinforcing material in mortars is shown to improve energy efficiency, improve ductility, and reduce waste. This tedious and careful consideration of the little things could make a big difference. Thank you.
Fascinating. It looks like the majority of your data are acquired on what would be, let's say, single bricks. Can you comment a little bit on how the properties are going to change when you start looking at larger capacities, larger loads, bigger thermal and mass considerations that would be used in, let's say, actual construction?
Right, so especially in the field of civil engineering and materials science, understanding how small samples work are usually representatives of larger things. However, there's of course variability and it's not all homogeneous. So for future research, what I hope to incorporate in this process is making sure we understand how the variability of bigger volumes, bigger sizes, larger amounts of fibers in our mixtures could affect that variability. And hopefully it's not too much so that it can stay consistent in the future.
Thank you so much. Very interesting presentation. I think
Yes, so I come from a poorer neighborhood in Indiana. That's where I was born and raised. And unfortunately, the threat of waste and the threat of just pollution, tangible physical pollution in our environments is a very real issue. And it unfortunately tends to inhabit lower demographic areas, lower financially able areas. So hopefully, if we incorporate the use of waste in our infrastructures, it could not only improve our infrastructures and get that waste off the streets and out of our environments, but it could also help with reducing costs because waste is very, very cheap compared to newer produced products. So more financially feasible while also improving the state of poorer areas.
Thanks. Thank you for the presentation. My question is about the type of fibers that you use. Have you determined that denim would be the best type of fiber, or is this what you're starting with to get a proof of concept, and then you would test the specific different types of fibers that you would include?
Yes, so this project in particular, it uses denim fibers, and we chose to go with denim fibers for testing because of how available it is. Denim is one of the largest, if not the largest, produced textile in the world. So we hope to address the consistency and how common this textile is so that it can be easier used because more production equals more waste equals more accessibility to incorporating these things in our infrastructures. However, textile fibers in general, not recycled, but textile fibers in general are used, but they tend to be synthetic. So in future research, which is what I hope to do, I hope to look at different organic fibers and different hybrid fibers, meaning natural fibers and synthetic fibers in combination.
Thank you.
All right, thank you everyone. Thanks for having me.
All right, so now the judges are going to take 5 minutes to deliberate, and when they're done, I'll just announce the next speaker. All right, next up we have Sam King, a junior in pharmaceutical sciences, who will be presenting BAF 170 in neuroendocrine prostate cancer. Please welcome them to the stage.
Hi, everyone. Hi, everyone. My name is Sam King. Today I'm going to talk about neuroendocrine prostate cancer and BAF 170's role in it. So with prostate cancer, right, it's one of the most prominent forms of cancer in men. And as such, we have developed a variety of tools to treat this. And one of the biggest ways is we use things called androgen deprivation therapy. So it's a form of hormonal therapy. And right, so androgens are these hormones like testosterone that will go inside these prostate cancer cells and stimulate things like growth and other normal cellular processes, and basically contribute to prostate cancer being able to grow and survive. And so it makes a lot of sense, right, that we're basically just targeting these androgens from being able to go inside the cell and do their thing and help these prostate cancer cells grow. What can happen, though, is that these prostate cancer cells can become drug resistant. And so this is common, like with all cancers, right? And there's especially a subset of these drug-resistant cells called neuroendocrine prostate cancer. And we name it this because they have an high expression of genes that we normally associate with things like neurons and other neuronally related cells. What's interesting, right, is when we normally are thinking about cancer, we're thinking about mutations. But when we compare the genome of these neuroendocrine prostate cancers to regular prostate cancer cells, we don't see a lot of mutations in these. So, right, the genomes are largely identical. And the thing that's really changing in between these is gene expression levels. So we think this is mainly an epigenetic process, which is just things that are not changing the physical structure of the genome that are changing gene expression. One of the proteins that mediates these kind of changes is our chromatin remodelers like BAF 170. And so what they'll do is, right, our DNA is not just floating around inside of our cell. It's wrapped around these spherical proteins called histones. And basically, it'll move these guys around to change gene expression. Why we think BAF 170 has a role in this process is that as these prostate cancer cells go into this neuroendocrine state in the lab, we see higher expression of our protein BAF 170. When we change the levels of BAF 170 in these cells, like increase it or decrease it, we notice that they either have higher, when we increase BAF 170, they have higher expression of these neuronally related genes. And when we decrease expression of BAF 170, they have decreased expression of these neuronally related genes, both at the RNA level and at the protein level. Why this is important, right, is that if we can basically if we could create a drug that could inhibit BAF 170, now that we know that it plays this role, we can basically cut off this neuroendocrine differentiation, which will kind of act like cutting off an escape route of this cancer. And then on the right, this is kind of what these cells look like, right? And so we can also, when we change levels of BAF 170, they look more neuronal, right? They have these longer kind of spindly outgrowths and smaller cell bodies. And when we decrease BAF 170, we see the same. Thank you.
All right, thanks, Sam. Can you expand a little bit on if there are any known functional redundancies, in the sense that if you were to develop a robust agent to be able to compete with BAF 170 or degrade it or stop its activity in some way, what other mechanisms might still be in play in cells that could still lead to this differentiation?
That's a good question. Yeah, right. We're not saying that BAF 170 is the only thing mediating this process, right? There's going to be lots of other factors that are, and I think there's lots of other epigenetic players that are in this process. But we just think BAF 170 is one of those and could possibly help in some cancers with this process. I think the best determinants would be looking at BAF 170 expression, and also there's this isoform of BAF 170 that we look at that is really prominent in this kind of drug resistance that we see in the lab. So kind of using the BAF 170 expression and its isoform expression.
Fascinating presentation. Thank you. I wanted to ask how what you found so far in your research differs from previous studies that have been found, or converges with other studies and other work recently published or ongoing.
Yeah, that's a great question. Yeah, so a lot of the stuff that we found, like about the epigenetics, right, is kind of being confirmed by a lot of different groups. There's kind of consensus that this neuroendocrine differentiation is largely a epigenetic process. What's different about this work, right, is that we're identifying a specific player in this process, which is BAF170. And then also that isoform that I was talking to Dr. Lonam about, that's also something new that we don't see in normal cells, and it is something special about this project.
Thanks.
Thanks.
Thank you, Sam. So with this project, what would be the next step that you would take in this project, and where could you go? But then how would other people potentially use this to continue this type of research?
Yeah. So one of the big ways that we're going now is, as I was kind of saying, developing a drug. So right, we've identified kind of a targetable druggable site that another group has identified, and we're trying to figure out if we can find a compound that can target this that we can maybe use in clinic or something like that. As far as work other people could do, it is kind of the same thing, and also just understanding the role of BAF170 in biology other than just prostate cancer. Thank you.
Thanks, Sam.
Awesome. Thank you, guys.
All right, presenting next is James Rooney, a senior in pharmaceutical sciences, who will be presenting the role of methylation in the protective activity of HSP31. Please welcome them to the stage.
Hi everyone, my name is James Rooney, and my research project focuses on studying the stress response of a protein called HSP31 in Saccharomyces cerevisiae yeast, which is considered a model organism for the human genome. HSP31 is a heat shock protein, which means it helps the yeast respond to cellular stress. HSP31 is also a parallel gene to the DJ1 gene in humans. The dysregulation of DJ1 in humans is involved in cancer progression and Parkinson's disease. If you think of a protein like a string, it has two ends. One end is called the N-terminus, and the other is called the C-terminus. Proteins are composed of molecules called amino acids, which form codes throughout the protein. Certain codes will tell an enzyme called the methyltransferase to modify the N-terminus of the protein through a process called methylation, which is the addition of one or more single carbon-based chemical groups to the end of the protein, as depicted in the top left picture. The bottom left diagram shows how single amino acid substitutions on the N-terminus of the protein can result in altered levels of protein methylation. We hypothesize that the N-terminal methylation of HSP31 will affect HSP31's protective activity against cellular stress. We study this process through a series of experiments in which strains of yeast are genetically modified to affect their methylation status. We also artificially induce the overexpression of HSP31 in order to study its protective activity more specifically. For example, in one strain of yeast, we delete the GLO1 gene, which is involved in the detoxification of a compound called methylglyoxal, or MgO. We also established that GLO1 deficient yeast, or GLO1 deletion, is sensitive to cellular stress by MgO. We then use mutants of various levels of HSP31 methylation to determine if methylation affects HSP31's protective activity against cellular stress. The results on the right show that HSP31 overexpression successfully rescues mutants in the GLO1 deletion background because cells with vector, which do not contain HSP31, did not grow under MgO stress, whereas cells with wild type, which do contain HSP31, did grow under MgO stress. The fluorescent bands in the image below show that the HSP31 protein levels were equivalent in all strains. These results also show that the A2D mutant was less sensitive to MgO stress, while the A2P mutant was more sensitive to MgO stress compared to the wild type. Because A2D is known to decrease methylation and A2P is known to increase methylation, these results suggest that methylation does play a regulatory role in the stress response of HSP31. Further investigation of mutants using additional methods will better establish the role of methylation in the protective activity of HSP31, which can then be applied to the stress response of DJ1 in humans.
Thanks, that was great.
Thank you.
I've always been a little curious in just thinking about how such a small modification, particularly of a very nonpolar group like methyl, can have such dramatic consequences functionally and mechanistically. Can you kind of comment a little bit on how you might think about how such small nonpolar modifications play such big roles and have such big consequences?
Yeah, so the enzyme that methylates the N-terminus of this protein identifies a canonical motif, which is XPK, and so the X is what could be modified, and X could be alanine, proline, serine, any amino acid really, but certain ones will enhance methylation status or detract from it.
Great. Thank you very much. Okay, my question has to do, I heard you mention that the yeast under discussion here specifically has a strong resemblance to the human genome. I'm curious to hear a little bit more about how what you learn about methylation and HSP, how this might be used for to essentially affect human quality of life. Kind of, where might this go?
Right. So HSP31 is what's called an ortholog or a parallel gene of the DJ1 gene in humans. And so you can assume that most of the protective activity of HSP31 in yeast will be easily transferable or applied to DJ1 in humans. So right now what we're trying to do is build more of a fundamental understanding of the biological process that underlies HSP31's protective activity, and then hopefully we'll be able to apply that to the protective activity of DJ1 in humans in diseases like cancer, Parkinson's, and there are other applications too, like diabetes or just aging overall.
Thanks.
project in general and thinking about your understanding of the background with this project, but whenever conducting your research now, did anything turn out that was surprising to you or something that you did not expect? Or if it wasn't surprising or something that you didn't expect, why? And how did that match what the background suggested?
Yeah, so in this experiment specifically, because GLO1 is known and proven to detoxify MGO, it makes perfect sense that if you delete the GLO1 gene, those cells will be sensitive to stress by MGO. And it's not shown up here, but right now we're working through a high throughput screen of, so up here you can see 5 mutations in one specific background, and this high throughput screen is in 1,000 mutations in a huge variety of backgrounds. And recently we just had a hit that we don't know the identity of yet, but it's being sequenced maybe as we speak, and we're hoping to find the results of it next week. And so that is a super exciting thing that we weren't expecting to find, because similar to HSP31, or it may even be HSP31, it helps the cells, including surrounding cells, combat the stress that the cells were fighting.
Thank you.
Yep. All right, thank you, everyone.
Up next is Carl Russell III, a senior in biomedical engineering, who will be presenting The Analyte May Be Hard to Detect, but we're going to stick to it, biosensor development. Please welcome them to the stage.
Good evening. 1.3 million people in the United States live with type 1 diabetes. 21 million live with type 2 diabetes. One of those patients might be your grandfather, a partner, or your child. Those same loved ones probably use a pinprick glucose monitor. They will it will require you to stop, sit down, take your pack out of your pocket, prick your finger, and wait for a result. It's painful and time-consuming. But there's some hope. There's a new type of medical devices coming out called a continuous glucose monitor. And these devices are coming out in force. Some examples include the Abbott Freestyle, Dexcom, and Eversense. These monitors are extremely easy to use. It deploys right on the side of your arm, and you can connect to your phone via Bluetooth. It sounds amazing. But what's missing? They don't last forever. My name is Carl Russell, and in the Laboratory of Implantable Microsystems Research, we're developing new methods to extend the longevity of these sensors through the development of anti-fouling methods and novel bioadhesives. We work with amperometric biosensors, which detect molecules by reading the current produced by the electrons resultant from the oxidative reactions. We make the sensor specific by attaching an enzyme that reacts with a molecule of interest. In this case, it's glucose, and we use glucose oxidase. If you wanted to detect acetylcholine, we use acetylcholine oxidase. And these sensors are extremely small. So I have to package them in the lab to make sure that they run on a computer. But once I get that down, we can start running some experiments. One of those experiments being coronal amperometry. This is a change of concentration in a solution that you manually do in the lab so that you can experience a change in current density on the computer. In a lab setting, this looks like a staircase pattern. To optimize this, we implement anti-fouling techniques after a contamination buildup, usually with bovine serum albumin. This uses a special method to shake the contaminants off the biosensors so that we can increase its longevity. To make sure that the sensor stays in place after we develop the bioadhesive, we will produce a bioadhesive, but when we have it in the lab, it starts off as brittle and dry. But after one places it in the place that you want and hydrates it, it becomes very strong, develops covalent bonds, and interacts with the amine groups that are present in a lot of tissues in your body. It is the hope that this research contributes to the improvement of biosensor manufacturing and biosensor applications. I think continuous glucose monitors are the future of diabetes monitoring, and if we have a contribution to that type of research, and these devices will come out, and they'll be incredible. And I think that'd be pretty sweet. Thanks so much.
Awesome. Thanks, Carl. Maybe you can clarify for me, in trying to understand some of the chemistry I think you're suggesting is happening, what about the side reactions that can happen with your anti-fouling agents? So something like BSA is a simple globular protein, but has lots of reactive sites. So reactive amines and other sites. So what about side reactions that might also happen on the surfaces or related to the anti-fouling agents themselves?
So what was observed in the sensors that we contaminated was that if we place the sensors in a solution, BSA will just clump up. It'll clump up together, and it'll clump up on a surface it can attach itself to. And one of the surfaces is the platinum surface of the biosensor. And when it clumps up like that, we see changes in effects of how well those oxidative reactions progress. So when we see that, the current readings, they get depressed, and it doesn't react as quickly as it would when it's not contaminated. Does that answer your question? Sure.
Very excited to hear about the cyborg developing elements that we're talking about here. My question, I guess, from a scientific novice perspective, you're talking about the device developing covalent bonds and sort of becoming better here and discussing extending longevity. My curiosity then is if they are more firmly secured but not permanent, what is the process then like of extracting something if it is more firmly secured but perhaps not permanent?
That's an amazing question. I didn't have time to talk about it in my research pitch, but these covalent bond reactions are able to be decoupled if you inject some triggering solution. So that triggering solution, when you apply it to that bioadhesive, it'll detach fairly easily. But if you don't, it'll stay pretty firm on that tissue. And our goal with that is to make sure that if we try to lift up on this thing, it tears the tissue, not the adhesive. And what that means is that the adhesive is doing its job properly, and that the adhesive is the one that's going to be failing. So good failure on the it's good if the tissue fails, bad if the adhesive fails, because if the adhesive fails, we're not doing our job.
Okay, interesting. Thank you.
Thank you, Carl. Now I just have a picture of people just ripping out thinking about the tissue and skin. So that's a wonderful.
Sorry about that.
But my question, so you were kind of getting to this, and Tara asked me this question, but my question is about the goal. So you talk about that it doesn't last very long. I know people that use this and having to go through the process of putting them on and taking them off, potentially not in the most optimal time. So what would be a goal of achieving something that is long enough that would meet what you're looking for, but doesn't require a lot of money to have something that is permanent?
So that's tough. Oh, sorry.
That also contends with the antibiotic properties and the longevity of that.
So if I understand your question, long enough is a tough question to answer, and it's a question that comes with risk. So the longer you keep something inside the human body, the higher risk it has of the body reacting to it, of it getting infected. So it's a multifactorial in the considerations of how long do we want it to be in there. The goal is, as long as if we want to implant it for a certain time frame, we want it to monitor consistently and reliably, and it shouldn't develop too much contamination on it. And so that's the overall goal. Does that answer your question?
So more about just the action itself and meeting its purpose as opposed to a specific duration.
Yes. Yes, at this point, yes. But the longer it's able to be implanted there and it doing its job, I guess the better.
Thank you.
Well, thank you so much.
And finally, we have Yufan Zhou, a junior in mathematics who will be presenting Challenging the Accuracy of Langevin Dynamics as an Approximation for Chemical Reactions. Please welcome them to the stage.
Hello everyone, my name is Yufan. It's a bad idea to use Langevin dynamics to approximate an uphill process of a chemical reaction. Sorry? Okay, it doesn't matter. Let's move on. Langevin dynamics is a mathematical tool used to simulate the stochastic behavior of particles in a fluid environment, which takes both deterministic behavior and random factors. An uphill process in the chemistry is a reaction or a process that involves the reactants with lower energy than the product, and thus requires the energy input to occur. A chemical Langevin equation is a popular mathematical tool to simulate the stochastic dynamics of a chemical reaction in applied mathematics. So here's the snapshot of my research. The first row is the chemical reaction for the real situations, and the second row is the Langevin dynamics, which is the simulation. On the first column and the second column, every trajectory is a process with blue starting point and red ending point. If the trajectory is closed, then it means it's a periodic orbit. The second column is a zoom in of the first column, which only focuses on the uphill process. The third column is slightly different from the other two. It's a line chart which describes the relation between the so-called least action and the initial momentum. So from these graphs, you may notice why Langevin dynamics is a bad idea in approximating the chemical reactions. Well, you may notice the enclosed area given by the trajectories in Langevin dynamics, it's much larger than those in the chemical reactions. Similarly, you may notice in Langevin dynamics, the least action in the Langevin dynamics converges to 0.25, while this value in the Langevin dynamics is approximately 0.013. They differ by more than 10 times. To see their relation, this research has proved that the enclosed area is equal to the least action value. It was proved by yesterday. It was proved yesterday. What does it mean? It means some mathematical assumptions are violated. So currently, this research is going somewhere deeper to figure out how Langevin dynamics failed to approximate the chemical reactions in analysis. This research underscores the importance of a collaboration between mathematicians, engineers, and all experts in order to advance our understanding of a complex phenomena. Any questions? Thank you.
Excellent, thank you. So I was hoping you could clarify two things for me, please. So the first is, are you thinking only of bimolecular collisions in what you're thinking about here? And what is the influence of two-dimensional versus three-dimensional constraints on the kinds of collisions that are happening?
Genius question. This research comes from a class of biomedical reactions. So this property will apply to a broad class of biochemical reactions. That's what this research is doing. It's limited to the length of this research, so I have to cut off a lot of details, but the conclusion also applies to more reactions. As for the 2D dimensions and the 3D dimensions, well, things would become much more complicated because I wouldn't want to introduce any mathematics theorem here, but I want to give you a conclusion that in the 3D dynamics, we will meet more complicated phenomena. For example, the trajectories will become a mess, will become very chaotic. That's in short. Okay, let's see what is next.
Thank you. My question is, so as I understand it, Longevin dynamics are currently used kind of as a rough model of these biochemical processes. And you're saying this is a bad idea, it's too rough a model to be used. I am curious, A, what sorts of biochemical processes the Longevin dynamics are used to model, and B, if you're saying essentially it's too rough a model to be used, do you have ideas about other heuristics or mathematical models that you think should be used instead? So what would a better model be?
Nice question. For A class of enzyme reactions in the human body could be used in a class of enzyme reactions in the human body applies this research outcomes. For the second question, we have investigated much more methods than Langevin dynamics. For example, you might heard about Monte Carlo method, which just spreads infinitely many points in a paper to count how many of them drops into a required region. That's a Monte Carlo approximation. And more methods is like use Markov chain method to include more randomness to improve the accuracy of the Langevin dynamics. Well, in addition, Langevin dynamics only does bad in uphill process. It behaves good in a so-called downhill process. Well, it seems we are run out of the time. We are sorry for that, but yes, I think I have answered all your questions. Thank you.
Thank you.
Thank you for your presentation. I have a question, but I'm going to come at it as someone in society that is a taxpayer. And so thinking about your research, what are the next steps, or how can this research be applied in ways that would benefit society, or what are ways that I can see the outcomes of the research that you're conducting in practice with companies or in applied research?
Long story in short, it will be a very dangerous step if we directly, or I can say, if we only apply the Langevin dynamics to approximate a chemical reaction with deterministic energy barrier. So that's a long story in short. That's what I can bring to the society. If I'm an engineer, I would be after reading this paper, I would be very careful when simulating a chemical reaction with energy barrier. So I have to apply more models to take advanced models to improve the accuracy. I'd better not to use Langevin dynamics in experiments or theoretical simulations and so on. I would, okay.
Yep, you can keep going.
I still have 10 minutes. Okay, that's good. I still have 10 seconds. I still accept more questions. If that's it, thank you for all of you.
Thank you so much for listening to all of the fantastic research pitches. And a special thank you to the presenters who put in such hard work to prepare these presentations. So the judges will now spend some time deliberating and picking our winners. And while we wait, you as the audience have the chance to vote for which research pitch was your favorite. So you should have cards with different options. So please circle the one that was your favorite. And if you are here for extra credit, just make sure to write your name on the card and we'll come around and collect them in a little bit. If you don't have a card, can you let us know? Is there anyone who doesn't have one? No? Okay, great.
Can Fowler Hall hear me?
Yes, JJ, we can hear you.
Thank you, Catherine. Can that be turned off so that the judges can talk?
Oh, here. I can collect the.
She just transferred. Oh, she's awesome.
All right, thank you so much for your patience. The judges have concluded their deliberation, and it is now time to reveal the winners. So when your name gets announced, please come up on stage and get your certificate and award and take a picture. And then we ask that all of the contestants stay after all of this for a group picture, and we have things for all of you as well. So firstly, the audience favorite, as decided by your vote, is Anaya Edwards. Next, our third place award goes to James Rooney. Our second place award goes to Carl Russell III. And finally, first place goes to Anaya Edwards. Please give a big round of applause for our winners and everyone who presented tonight. And can all of the presenters please come on stage for a group picture? All right. And then
Thank you, everyone who attended this event tonight. Thank you to all the presenters for their amazing work. Thank you to the audience for showing up and supporting everyone, and thank you to all the judges for their amazing insight. Special thanks to the Student Organization Grant Allocation Board for funding this event, and thank you to the Hall of Music Productions for all the AV and technical support tonight. Thank you, everyone. We hope that this event has inspired you guys to go out and make your own discoveries. Have a great night. We'll see you guys next year.